Welcome, everyone, to our December coffee chat. I'm Laura Knutson-Murray. I'm the GIS coordinator and community outreach assistant with IrisBG. And with me today on the call is Shanna Jones, who's our social media and community outreach assistant. The recording, presentation, and other material will be made available in our Knowledge Library. All of our previous coffee chats can be found there in the category Articles and Presentations. You can also follow the topic IrisBG Coffee Chat and be notified by email every time something new is published. The idea behind these sessions is that we at IrisBG would like to offer you a space where you can connect with your peers, have a chat about IrisBG, and issues that may come up with the work with your collection. We don't have a fixed format for these, so please get in touch. If you would like to present on a challenge or talk on a topic, it can be anything big or small. Hopefully you will make some new connections today, and together we might become a little bit wiser. Save the date. Our next coffee chat will be in February. It will be given by Melissa Kalina from the Coastal Maine Botanical Gardens. She will talk on conservation threat analysis, so be sure to sign up for that. This map is an overview of the institutions that registered today. It's good to see so many of you from the community. And then we will launch our poll where you rate your experience using IrisBG. What is your level of expertise using IrisBG? Are you trying out the trial, a novice, intermediate, or advanced user? Thank you for participating in that. Our agenda today, we'll hear from Shari Buckhout from the San Diego Zoo and Wildlife Alliance. She has been a plant registrar for 35 years, so she's a IrisBG super user. So pepper her with questions, and we'll have Q&A at the end. So I'll turn the controls over to you, Shari.
Good morning. Hello, everyone. Thank you for having me today. As Laura said, I'll be talking about how the San Diego Zoo and our sister campuses have transitioned to IrisBG and are using it to further management of our plant collections and conservation. I have been working at the zoo for 35 years in the same position as plant registrar, during which time I've had 5 offices, worked under 4 curators, maintained and developed 5 plant record systems, and seen many, many changes over the years. Although I'm the only plant registrar, I've managed light-duty personnel, work-study students, interns, and volunteers, and I've trained many staff members for most of my tenure here. Every plant tells a story of conservation, innovation, and global impact. At SDZWA, our records are the foundation for saving species and habitat. But first, the history. The San Diego Zoo was founded by Henry Wegeforth in 1916. It is renowned for its innovative open-air exhibits and conservation efforts, housing over 12,000 animals from more than 680 species and thousands of exotic plants. The decade of 1926 to 1936 saw the planting of more than 5,000 trees from all over the world. Their larger tree planting holes needed to be blasted with dynamite to get through the thick bedrock or hardpan layers. But it was the decade of the 70s that brought the zoo the recognition of what it was, a zoological garden, with the establishment of the first irrigation systems and the first plant collections. Every plant tells a story. Doing some historical research over the years, we discovered this image going all the way back to 1915 during the exposition, where there's this tiny little sapling growing in the left-hand corner. From a sapling, Pinus mugo, in the Japanese teak pavilion to the original plant seen here in the Wildlife Explorer Base Camp, which is a renaming of our children's zoo area. It's one of the oldest plants still alive at the zoo today. So fast forward to 2025, the Zoological Society receives its American Museum accreditation. At that time, only 5 other zoos were accredited. That started in 1993 for us. Both the zoo and safari park are also recognized botanic gardens in Arboreta. And just last year, we were honored by the American Public Gardens in Arboreta to receive the Garden of Excellence Award. We represent about 700,000 plants at the zoo alone, including over 13,000 prominent specimens of over 3,000 species. Here are some images that I wanted to include that represent the safari park at the bottom left-hand corner. You can see it's in a much more temperate area than us at the zoo, who are more coastal. And I wanted to give a shout out to our newest exhibit opening called Elephant Valley at the Safari Park. That will be open March 5th of 2026. So please join us. Metrics. The San Diego Zoo's horticulture department. Also over 1,900 acres of growing space when the safari park is included. 400 in active maintenance. 1,500 dedicated to preservation of natural habitat and habitat restoration. You can see here we have a staff of 40, which includes horticulturists, arborists, propagators, plant registrar, myself, browse team, collection experts or enthusiasts. We are utilizing 4 greenhouses, 2 of which are orchid greenhouses, a propagation greenhouse, a carnivorous plant greenhouse. And in 2005, we started developing our micropropagation lab for our conservation work with exceptional endangered species, as well as a seed bank, which we also house the biobanks for the native plant material. We have 8 accredited collections seen here, as well as some special collections of note, including Hawaiian plants. We have 5 Hawaiian gardens actually now, going on 6, a caudiciform collection, carnivorous plant collection, and a very nice hibiscus collection. And you can see some of the stats here from our IrisBG generated reports. We really began to grow our collections between 1976 and 1986 when we started doing collecting trips to Papua New Guinea and came back with lots of salvaged orchids from timber areas under harvest. And then in the 1980s, our bamboo collection really expanded with the introduction of giant pandas. The San Diego Zoo Wildlife Alliance exemplifies conservation-driven horticulture, focusing on direct science, education, and outreach. We partner to preserve rare and endangered plants from many biodiversity hotspots. Our team efforts include seed collection, tissue culture, cryopreservation, and sharing propagation protocols with partners across the U.S. and Europe. You can see that we are a team plant made up of the San Diego Zoo and the seed bank, which is held at the Beckman Center for Conservation Work or Conservation Research, the Safari Park, and CPC. Plant records fuel conservation success by seed banking, tracking endangered species from several of our collections here, sharing data with global partners to prevent extinction, and having real-world impacts. Our conservation efforts focus on exceptional endangered species, otherwise known as EEP. These can't be banked through traditional means, and they require direct scientific research. For example, oaks, the acorns cannot be seed banked in the traditional way because they only last about 6 months to a year, and their viability requires a high moisture content that freezing and drying destroys. With orchids, they have microscopic seeds that require fungal interactions in the environment to germinate and grow. And cycads, one of the oldest plant groups, the seeds cannot be dried and frozen, and currently the best storage system is a living collection. So first and foremost, we're trying to end extinction in our own backyard. We're partnering with the GCCO, Global Consortium for the Conservation of Oaks, to preserve oaks worldwide, focusing on 6 priority oak species from Southern California and the Channel Islands, the U.S., Baja California, and into Mexico. We collect acorns, grow seedlings, share with our colleagues, and create safe sites to preserve genetic populations for future conservation work. This is a particularly lengthy and delicate process. One in particular, the Newtowns scrub oak, is endangered by the IUCN, and non-seed material has been remarkably cryopreserved in our zoo's frozen zoo oak localities. I've created here a PCN report with saved filters, such as this one for our oak collection. You can see that our records now include many entries with locality data that occur throughout Southern California, including the Channel Islands. Here's an example using the item history report, which I've used to audit our oak propagation records, especially when we transition to item numbers such as 101 for the seed bank, 102 and above for propagation item numbers. To finesse oak conservation, we micropropagate tissue from viable acorns, make the little forest babies, freeze their shoot tips and somatic embryos in cryopreservation tanks. Then we thaw, see if the tissue is still viable and able to be rooted. We will ultimately endeavor to hammer out this science behind how this works and publish and share the results. Another area of biogeographic focus is in Palau, which has the highest biodiversity of orchids in Micronesia. Our plant conservation and horticulture teams helped many Palauans acquire skills in plant propagation techniques for their unique orchids, including collecting seeds from the wild. We have been successful in propagating the Micronesian cycad from wild collected seeds as well. Our excursion started in 2015, and then we revisited in 2022 and into 2024. We were able to successfully propagate many of them in our propagation lab, and some were sent out across the United States and Europe for ex situ conservation. Here you can see some of the beautiful photos of those orchids and even an image of a Palauan stamp that includes a native orchid. In 2024, when two of our staff members, Sawa Lagani and Heinfried Block, went on the next excursion to Palau, they were able to breed back seedlings of Lypodium and Dendrobium in vitro back to the place of origin, returning those propagules to Lake Nardoc Nature Reserve, where the seed was originally harvested. Here are some images of how they looked coming out of the micropropagation lab and into potting soil in our greenhouse, or actually in their greenhouse as well. This is the one that they reintroduced, Bulbophyllum hatusemanum. Oh, that's a hard one to say. So during our excursions, we found mature seed capsules of this rare endemic orchid species. And two of the gentlemen that came to the San Diego Zoo for training, MC and McKnight, local Palauans, they germinated on chosen media that turned out to be very good, very successful. So we are currently assisting in raising the plants via regular communication and video calls with them. If you don't know about Palau, here's an image of where it actually occurs. It's nestled between Australia and Japan, the Philippines, and Hawaii. It's an archipelago of 8 large islands, 12 intermediate islands, and over 500 rock islands. It's been interesting to learn about this region and challenging to decipher the collection data into neat packages of tabular locality in IrisBG. As a note, receiving this information like locality data is often quite challenging, as you probably know too. Oftentimes, I'll get information from our horticulturists in a plethora of ways, including direct conversation, emails, GIS projects, and spreadsheets. It takes some time to sleuth out all of the locality property fields data. For example, I may only get the subdivision 1 and have to look up the country, or get the subdivision 2 and have to look up the subdivision 1. And sometimes I only get the coordinates and have to look up all the rest using GIS and the internet. Another one of our conservation projects is about the Pritchardia palms, or Lulus. In 1979, our horticulturist Ernie Chu was given the opportunity to receive seed from the Pacific Tropical Botanical Garden, which is now NTBG, the National Tropical Botanic Garden. We received seeds of the endangered Pritchardia Ka'ale variety minima from Oahu. Only 30 trees remained in the wild at that time, and only 10 were mature specimens. When I first started in 1991, the old propagation record was found here on the left, and then 3 successfully growing specimens were located on grounds, pictured in the image in the center, and then finally tied back to the original data of the correspondence. It's like finding treasure being a plant registrar. To amplify our conservation impact in Hawaii, we are partnering to conserve the threatened Pritchardia. Together, our partnerships collectively support the recovery of all 24 recognized Pritchardia species across the Hawaiian Islands. DOFA, part of the Department of Land and Natural Resources, DLNR, has a rare plant program that protects wild populations through seed banking, greenhouse propagation, outplanting, and population monitoring. We started traveling to Hawaii in the early 2000s and received our first seedlings as part of the new agreement for ex situ conservation collections, including 3 Pritchardia bakeri and 2 Pritchardia kahukuensis. There are around 25 species in tropical Pacific islands altogether, and as many as 19 endemic to Hawaii alone. Its main threat is the coconut rhinoceros beetle, which was first detected in Oahu in 2013 and then has moved to additional islands by 2023. It unfortunately feeds on the palm hearts and emerging palm fronds and is very devastating. At the San Diego Zoo Wildlife Alliance, we are dedicated to protecting plants and wildlife worldwide. By generating, sharing, and applying scientific knowledge vital to the conservation of animals, plants, and habitats, we contribute to the global fight against extinction. Here's an area of the IrisBG that includes data on the permitting process and phytosanitary certificates that we needed to employ in conserving this palm. In 2022, one of our horticulturists, Andres Espinosa, traveled by helicopter over Oahu just to hike to the remote location of the regional group of Ka'ale. It was quite a trudge through thick shrubs and a lot of enormously large ferns, from what I understand. Here you see a few of our collection enthusiasts, propagationists, and our horticulture manager processing these rare acquisitions in our seed bank and lab facility, as well as the nursery. Pritchardia martii was one of the first ones that we had success with growing on grounds here at the zoo. We extracted the embryo from a green seed, and a number of years later, here on the right, we have 2 maturing Lulu martii in the first Hawaiian garden. Why a modern record system matters. Today, our plant record system in IrisBG unifies data across the zoo, safari park, and Beckman Center for Conservation Research. This integration means every plant, from rare orchids to historic oaks, is tracked with precision, supporting conservation research and daily garden operations. A modern database replaces fragmented legacy systems and paper logbooks, eliminating inconsistencies, enabling strong reporting and taxonomic accuracy. Our staff can quickly access, update, and analyze plant data, saving time and reducing errors. Our system supports complex workflows like seed banking, tissue culture, and permits, critical for conserving endangered species and managing multi-continental collections. It enables us to collaborate globally and respond rapidly to the conservation needs. With dashboards and custom attributes like harvest and pollination dates, our staff can visualize collection status, track propagation success, and streamline reporting for better decision-making. We've also included monthly meetings and shared protocol to ensure staff are trained and onboarded efficiently, fostering a culture of continuous improvement and teamwork. So every plant story is recorded and accessible. All right, a review of our plant record systems. Since I started here in January 1991, about 35 years ago, we have gone through many plant record systems. Databasing was new to me, and training was simply handing me the keys and telling me to figure it out. So you can see I did. We went through a few iterations of software and applications, starting with an AS400 system similar to that of a mainframe, and then to versions of DBase, Access, and finally into IrisBG. From the 1970s to 2012, handwritten logs and accession cards were the norm. We phased out written logbooks, of which we were still missing 10 years, in 2012, and accession cards in 2020. What started out as a browse and toxicity database for the Los Angeles Zoological Gardens was built with the help of many horticulture specialists, students, personnel, volunteers, and Jenica Jones, who was a horticulturist at that time. This is what some of those screens look like for that system in Access. IrisBG, the engine behind our collection. It unifies fragmented databases, centralizes management of plant data for the zoo, safari park, and the seed bank. It links our accessions, items, propagation, herbaria, locality data, collection data, permit, archive documents, images, and maps. It has a strong taxonomy, management, analysis, and reporting system, as you probably all are aware of, and it's ideal for conservation research and garden operations. Growing pains. I'm sure all of you had growing pains, and I don't mean to be negative, I just want to commiserate. Across 2 campuses and 3 facilities, we met monthly for a year to the ultimate purchase of IrisBG. We discussed many database options, reviewed our data similarities and disparities, and agreed on the best shared solution, IrisBG. Over that year, leading to the ultimate purchase of IrisBG, we discussed database options and reviewed our shared data similarities and disparities. So I created this field definitions spreadsheet, which required querying data from the Access system that we currently had, their quantities per field, and determine which that needed cleaning before the transition, which that needed to be eliminated, and which could be parsed out. I identified the ITF fields, query data quantities. So some of the keywords that I looked for and to do this cleaning was things in our comments fields like keep, modify, convert, audit, remove, and maybe questionable. And that's how we made our determinations on what fields we were going to keep. All right, this is what it ended up looking like. All of the families, taxonomic information, and accession and items information. So through the zoo, the safari park, and the native seed bank, pulling from Access databases and an ArcGIS data exports, we ended up with data on taxa, accessions, and items, plus 715 locations, over 1,000 contacts, 71 library records, and almost 2,500 localities. We merged into a single database, made up of the safari park, the seed bank, herbaria, and the zoo. Since the safari park used to be called the Wild Animal Park, their accessions still begin with W, hence the name Wild Safari Park here in the status field, which we have now changed to Safari Park. We've also added an M status for multiple sites when we share an accession. And grandfathered systems. We all know what a challenge those are. We had been using an alphanumeric system of identifiers that we had to translate and standardize to improve our traceability and reporting. As you can see here, many of them translated into a high number of records. And in that translation, they were saved in Conserved 1 and Conserved 2 fields in IrisBG. And many of them were given high item numbers, which were a little bit confusing at first. Post-transition to IrisBG, we inherited many event-driven data cleanup tasks. These are systematic tasks that have addressed our taxonomic issues, including synonyms, deprecated genera, and species that weren't validated. Additionally, we ended up with thousands of unhoused propagation entries, most of which have been completed at this point, and open issues are being tracked for ongoing improvement. It was a major data cleanup, and this issue was particularly daunting. The current records were perpetuated down the historic levels, if that makes sense to you all. You can tell by the comments that it was still in a 5-gallon container in the propagation area, nursery 4. Those are being updated as we come across them. We had to look for key status words such as move, which over 1,000 records contained the word move, mapped, shifted, and divided, and then move those from the comments field to change the appropriate status fields and location. We have now streamlined our propagation data based on some great work with the coffee chat, particularly those from Kew Gardens. We had used propagation, including sow and strike dates and shifted history for each item. And now we've become much more efficient and succinct by moving them into propagation data items that are dedicated strictly to propagation. We've been digitizing our paper documentation for a few years now, and I'm very proud to say we've emptied a giant 6-foot-tall file cabinet with 4 drawers and a dozen or so project binders. Can you relate? As you can see by this exported report on localities, most of our wild localities are from the United States, reflecting the work of the Native Plant Seed Bank, as well as the zoo's work on southwestern oaks that extend into Mexico, and the Hawaiian natives, including collection areas for Pritchardia, the lulu palm. Data import templates. We have increased our rate of creating accession records with the use of data import templates. The initial IrisBG provided template has been partitioned here into multiple templates based on common compartmentalization of record creation activities, which eases our data import. Each is maintained as a read-only file due to frequent unintentional modifications. Multiple sheets are developed to support the creation of data validation lists per field/column. This improves the format and value lookup process. Examples here are of received person values based on the personnel table, accession type choices, and material codes. The import items template includes drop-downs for item locations, nursery areas only at this time because we have quite a few on-grounds locations by grid, the item status text, and propagation container types and sizes, as you see here. The field collection imports table doesn't offer drop-downs or data validation lists. Locality and collection fields are complicated and challenging at times. We've learned how to add custom attributes from a recent coffee chat as well. We have now data fields for pollinator, pollination date, harvester, and harvest date, as you can see here in this spreadsheet that was exported from IrisBG. Dashboards. Dashboards are handy for visualizing the dynamic collections at any one point in time, but we usually use them at the completion of a new project, exhibit area, or at the end of each year. I report on society-wide stats as well as zoo-specific stats. I've broken them down taxonomically as well as numerically, from simple reporting accessions added to breaking out those that most conservation impact per IUCN code and those that include wild collection provinces. Whether you use a pie or bar charts, as I have done here, I can enhance my interpretation of the data metrics visually for easier audience assimilation. We've been endeavoring to test our entire orchid collection to ensure a virus-free stock. As you can see this image on the left, where we emaciate some of the plant material and test it for virus. We've accomplished several large imports of data from the native seed bank, a high percentage of which are represented by the Asteraceae family. Additionally, we've increased the tabular data established of thousands of localities, primarily from North America continent, followed by Oceania. Through the use of Floria for field inventories on grounds, we have greatly increased our photo documentation of the collections as well. The reference files. Oh, I can't forget the reference files because this is what we've been working the hardest on over the past 3 years. The biggest increase in our plant records documentation has occurred via the process of scanning and attaching files to the reference files section of IrisBG. The second largest collection, palms, has included over 1,500 files alone. Lesson learned, though, after analyzing the description and file naming constructs, we find the need for protocols to keep the formatting more concise and contiguous for easy reporting and analysis. I'm sure you can all relate. Okay, from logbooks to dashboards, here is a snapshot of our data journey. From 1970s to about 2012, we were still using handwritten logs and accession cards. Then in 2000, we started introducing digital mapping. In 2019, when we acquired IrisBG, we added propagation, locality, collection, imagery, permits, and customized fields. From 2022, we started digitizing and linking our legacy paperwork and maps. Today, with shared import templates, reports, training, and real-time dashboards, we have come into the future. All right, so here is a snapshot of SDZWA's IrisBG environment. Again, I want to reiterate what we've accomplished. We've unified 3 databases across the zoo, safari park, and Beckman Center for Conservation Research. We've integrated our herbarium, seed bank, tissue culture, and propagation records. We have dealt with a high volume of collections with complex histories. So we've incorporated the use of custom attributes, import templates, and saved report filters. And we've archived legacy systems of accession cards and transitional documents and maps. From the AS400 and DBase to Access, and now IrisBG, San Diego Zoo Wildlife Alliance's plant records have evolved from paper logbooks and fragmented databases to a unified modern system. As you can see here, we've made many accomplishments that I'm happy to say is due to the use of IrisBG. This transition has resolved historical data inconsistencies and enabled strong reporting and taxonomic accuracy. I can't go without mentioning this new species discovery. We have a new Chamaedorea delicata for the palm family here on zoo grounds. Congratulations to our staff and volunteers and to Don Hodel for finding, documenting, and publishing this amazing work. And I can't wrap things up without a special nod and thank you to my dedicated volunteers who helped tremendously with inventories, accessioning, item updates, interpretive data development, tagging, mapping, and archiving. The list goes on and on. Thank you. San Diego Zoo and Wildlife Alliance's commitment to plant management drives new species discovery, conservation-driven localities, and ongoing data import expansion, ensuring the organization remains at the forefront of botanical excellence. Modern plant database management at SDZWA is not just about record keeping. It's about empowering every staff member to protect, manage, and celebrate our living collections. With IrisBG, we now have the tools to drive conservation, streamline operations, and collaborate globally. Every record, every protocol, and every partnership strengthens our mission and legacy. How can you use plant data to drive conservation in your own work? Let's collaborate to protect biodiversity, one record at a time. Thank you.
Thank you so much, Shari. That was fantastic.